participant. Myself, Dr. Amrin Sultana, holding the cadre of Assistant Professor at MLR Institute of Pharmacy. It gives me an immense pleasure and honor to welcome you all to the fifth day of our EFDP program on integration of AI and network pharmacy in modern pharmacological sciences. We have successfully reached the final day of this enriching 5-day journey. Over the past few days, we have explored diverse and emerging dimensions of pharmaceutical sciences, particularly focusing on the integration of artificial intelligence and network based approaches in modern pharmacology. Today's session marks the culmination of this insightful program, and I'm confident that it will be equally enlightening and inspiring. Let us begin the session with enthusiasm and a spirit of learning. Now, it is my proud privilege to introduce today's distinguished speaker, Dr. Vinyas Mayasa. He is a passionate teacher and researcher in the field of pharmacology and toxicology, currently working as an assistant professor at GITAM School of Pharmacy, Hyderabad. With over 15 years of experience in both academia and industry, he is known for making complex concepts simple and relatable for students. He completed his B.Pharm from Usmania University, M.Pharm from Rajiv Gandhi University of Health and Sciences, and PhD, where he worked on insulin metabolism in diabetes, an area that continues to influence his research today. His interests include anti-diabetic and anti-obesity research, neuroprotection, toxicology, and natural product-based studies, with a strong focus on connecting laboratory research to real-world healthcare application. Beyond his research, Dr. Vinyas is a dedicated mentor who enjoys guiding students at all the levels and encouraging them to publish and innovate. He has authored 49 research papers with over 240 citations, along with books and book chapters, reflecting his consistent academic contributions. He had recognized with the Best Researcher Award. He actively participates in conferences and faculty development programs, sharing his knowledge and learning from the peers. He is particularly interested in integrating modern approaches like in silico methods and critically explore new ideas and contribute meaningfully to healthcare and scientific advancement. We are truly honored to have such an eminent personality with us today to share their knowledge and insight. On behalf of MLR Institute of Pharmacy, I extend a warm welcome to you, sir. Without any further delay, I request our esteemed speaker to kindly take over the session and enlighten us with their valuable thoughts. Over to you, sir.
Good morning. Good morning, everyone, for the participants and the speakers, as well as the attendees who are here. And thank you, Dr. Amrin Sultana, as well as Dr. Murli for introducing me for this FDP, and really enthusiastic to participate in this event and to share my insights of the ethnopharmacology for the precision. Okay, Dr. Amrin, can I share my screen here now?
Yes, sir, you can share your screen.
Oh. Can you see the screen? Hello?
No, sir. No, sir. Not yet.
Okay. Is it okay, sir, now? Hello?
Yes, sir. We can see your screen, but not PPT.
Okay, sir. Okay.
You just open your PPT.
I opened the PPT.
No, we are seeing your Microsoft Teams
Teams itself.
screen.
Okay. In that case, can I, Murli sir, can I share the PPT to you?
Yes, sir. Please send your PPT to my WhatsApp.
Yeah.
I'll do that from here.
Yeah, sure. 1 minute. Yeah, Murli sir, I shared the screen PPT to you.
Yes, sir. Give me a
Yeah.
second.
Are you able to open this PPT, sir?
Yes, sir. I'm just downloading it.
Okay. You can just click on the presentation view, sir. Yeah. Yeah.
Yes,
Thank
sir.
you,
I
Murli
think you
garu.
can continue now.
Yes, sir. Thank you, Murli garu, for assisting me for this. Actually, I need to restart my computer whenever I had to make the changes in the computer. So, good morning, everyone, and welcome to the today's FDP presentation. Myself, Dr. Vinyas. I am working as assistant professor in GITAM School of Pharmacy in the Department of Pharmacology. Today's topic is AI-enabled translation ethnopharmacology for precision medicine. As we know that the herbs are available in our nature, so the point is that how we are going to utilize these herbs for the precision herbal utilization. We know the terms herbal medicine and precision medicine. Now the combination of herbal precision medicine is my real-time topic for today. So if I go for the next slide, that is contents of this. Next slide. So Murli sir, next slide. Murli sir, is it audible to you?
Yes, sir. Yes, sir. Now it is okay.
Can you scroll down to the next slide, sir? Yes, sir. Right. So now if you go, I think it's not open. So the contents of the slide is like introduction, phytochemical profiling, and how you're going to identify separation and identification of the compounds, and what are the challenges that are to be there for the quality assurance people who are working in the industries, and what are the emerging trends, and what are how the drugs and their actions are going to work. And the interesting part of my presentation is that we are going to do a real-time experiment here, what we have performed for the last 2 days in my lab. So we are going to explore that how we performed using the network pharmacology by herbal precision medicine. I will show the real-time data for you, how we worked out and how we can easily identify the mechanism of action of the herbal drug by using the network pharmacology. So here we have established the mechanism for the herbal medicine. That's an interesting part. And we are now in the yet to process to validate that mechanism by both in vitro and in vivo models. So, and finally, we are going to into the part of for the machine learning in the clinicals, where the PharmD people will be morely or they will be in the highly interested to see this presentation of how the machine learnings will be used in the clinical trials. So next slide. If you go for the herbal medicines, now we have been using for the centuries for the Ayurveda and traditional Chinese herbal medicines, and Unani have been using for the centuries for us, and it's a folklore for us and it is easily available. So in and even the WHO says that 80% of the global population is still utilizing this herbal medicine. So in our allopathic system of medicine, along with the herbal medicines, around 80%. So it's a good number for that herbal medicines are still being used. And now the question comes is that validation of these herbal medicines is an interesting part here. And if you go to the revenue part, the herbal medicine is projected to surpass 200 billion by 2030. It's a very good number. So there is a huge market for the herbal medicine. Now the thing is that how you're going to utilize this herbal medicine, how do you are going to authenticate these herbal medicines for the world to be used? Because as allopathic medicine practices, we know that there is a lot of side effects and adverse effects have been reported for these allopathic medicines. So to overcome this, precision personalized herbal medicines, because we already know these medicines have been used for centuries and decades, and every year centuries we are using these herbal drugs. Now the question comes is that why can't be you it can't be personalized to person. So now, even though it is used now, integration of science, that is scientific validation of these herbal medicines, is a crucial part, whether the plant is real or original or a duplicate, because their adulteration and some different kinds of compounds are added into that. So validation of herbal medicines is important. Now the question comes, the same plant grows in your mountains have a different phytoconstant, and the same plant grows in the plain areas go have a different phytoconstants. Now validation of the same plant it grown in the different geography, season and cultivation practices makes it different. So validation is an important thing for us. Now along with that, adulteration is one of the major criteria where the safety and reliability of our product is lost. Now you have to ask the question, as it is used for the human use, what are the human applications, the safety and efficacy and consistency across the batches, batch to batch, where the allopathy system are having a strong area of the scientific validation. Our herbal medicines are lacking these scientific evidences, and that's the reason we are losing the market in the world. So now going for the next slide, phytochemical profiling. What is phytochemical profiling? So now in the phytochemical profiling, we are going to purify the or identify the compound and analyze the chemical entity that is responsible for causing this drug. Now here it is also called as a fingerprint of the sample, where because every plant has a unique feature of having a separate chemical entities that are responsible for this action. So now by using the phytochemical profiling, we are identification and quantification of these compounds we are going to do. And along with that, we are going to authenticity. So we are going to confirm it that this is the product. I will say real-time experiment in what we did in our PhD lab. So in Ayurveda, the same plant is called by multiple names, or the multiple names are called by a single plant. So whenever you are having the texts having the written as Vinyas somnifera or some other plant that is used, but instead the plant is having the different names in different area. Now the question comes is, what is the authentic of this product? Ayurveda say use of Vinyas somnifera, but Vinyas somnifera is called by the different names, or with this is different kind of thing. So now the question comes, it is useful for our plant, but how you are going to authentic that this is the plant and this is the standardized value. So now we are going to do in that part also. So we are lacking in that area identification. This can be done by the phytochemical profiling. So next slide. So to address this one, we are going to do the phytochemical profiling by robust analytical methods are required. So the robust analytical methods like GCMS, LCMS, GCMS, and we can also use NMR for and other techniques that we can be using. So next slide, advancing phytochemical screening. Now we are going to do high throughput profiling. In the meaning high throughput profiling means, so at a time we are going to have millions of compounds that will be filtered throughout. By standardized method, we are going to establish and reproducible profiling methods will be established. And we are going to create a comprehensive database where this work should not be repeated by someone, and there should not be a waste of time for someone who is doing the same work. So we are going to create an extensive database of phytochemical profiling and their properties. In this area, we have a lot of research work, or there is a huge scope for the research.
Hello, ఎవరండి?
Hello.
హలో సో మామూలు కాల్ చేయండి, నెట్ కాల్ అర్థం కావట్లేదు.
And along with that, we are going to integrate the omics. So in the omics, we are going to identify the proteomics, genomics, and gene databases, gene banking, and other technologies. We are using that, and then we are going to identify the compound and the mechanisms as well. So now we are going to apply all these genoprofiling methods. So now separation and identification techniques. Next slide, sir. So in the next in this one, we are using, these are the gold standards that we are using. In this one, TLC will be used, thin layer chromatography. For our B.Pharm graduates, we are using this one, thin layer chromatography. It's a simple technique where with the RT levels and other RF levels, we are able to identify. And by the HPLC, we are able to identify and purify and quantify the compounds here. Gas chromatography can be also be done. Here, LCMS, GCMS, these are the fingerprint technologies for the plant identification methods. So now we are using both the HPLC, LCMS, GCMS and other mass spectroscopy methods for identification of the compounds. So now what are the challenges that are in there in the herbal drug regulatory quality assurance? So sourcing and authentication. If you go for the next slide, sourcing and authentication, the first thing is ensuring that the correct identification of the product is done, and that is a suitable sources, because we will get the same plant from the different sources. Now how you are going to identify? So in allopathic system of medicine and other our pharmacy practices, we will get a sample at COA, certificate of analysis of the product. But here in the herbal product, only a botanist will certify this is the plant. The authenticity is missing, where the the names are called with the different locations and different names. So, and which part of the plant you are collecting and sourcing is also very much important. And chemical stability, at what a time you are collecting the plant. Example, mangoes you can collect in the summer season only. If you collect it in the mid-winter or in the rainy season, the purity of the compound will be missing in that, or the standardized product of promangiferin that is mixing in the leaf extract. So now, and along with that, whatever the product you are taking, the consumer perception is also very much important. The consumer should be aware what he is doing and awareness of the herbal safety, because along with the allopathy, the herbal drugs are also are intended to cause adverse effects and side effects. A clear pharmacovigilance should also be addressed for this one. And along with that, you are going to do the standardization of the extracts. So now consistent herbal extract should be there, standardization should be there. And recently we got a project on this one, standardization of the product from one of the company sponsored that. So they are expecting that we meet our it is a multi-component product. We got it, and they are saying that to standardize our product and make it as an authentic, that this is the product that the patients are going to consume it. The customer is taking that, make it an authentic and make sure that. So for that, we got a sample from the company as well. And along with that, regulatory compliance, as we are exporting the herbal drugs from the different countries, so that's the reason you have to establish the safety and labeling regulations that are required, and how the patient should be using, all the things should be there, and the quality control, because it's an herbal drug, how you're going to packaging it, because as you are exporting and utilizing it for different locations and other things, the main ingredient of the drug can be lost. So now the quality control, quality assurance, all these processes are very much important for an herbal drugs. So now if you go for the next slide, that is the emerging trends. In the emerging trends, we have metabolomics, proteomics, where it is a holistic approach, where we are using the plant biochemistry as well as the therapeutic potential. Formulations, in the recent days, we have seen many of our young PhD students are doing on silver nanoparticles, zinc nanoparticles, gold nanoparticles, and other heavy metal nanoparticles for the drug delivery and drug delivery systems, and to improve the bioavailability of the drug. So they are purifying the original part of the compound, and they are adding up to the silver, and they are trying to deliver the drug into this, because the nanoparticles can be delivered into the location, and it is a targeted delivery. Now for us, for the new future generation, wherein we are stepping into the AI world, how we are utilizing the AI for analytical methods. So now predicting the model, big data analysis and rapid profiling. So in our recent study, we have identified what are the responsible enzymes or hormones that are causing the diabetes, and we have collected a huge plant database from our network pharmacology. From that, we have identified the plants that are corely related with the established mechanism. Without doing any laboratory studies, we could be able to identify because of the data available in the net. So now we have planning to develop a model. That model can easily identify the plant, whether it will be useful for anti-diabetic activity or not. So by this, we can be easily be doing the profiling of the plant and utilization of the plant. So these innovations will be real shaping the future of herbal research industry. Now, if you go for the next slide, that is opportunities and research and development. So now we, as herbals products, we have export potential. We can export the drugs to anywhere, and it is a growing international demand. as the allopathic system of medicine has adverse drug reactions a lot more. So the world is choosing for an alternative systems. The herbal drugs are the main component where there is a huge demand for that. So in one of the research committee, we were using the banana peel extract for anti-diabetic activity. Imagine banana peel extract were also be using, and that product was also in the market in the US. Right. So now in the next category, we are going to go with the personalized herbal medicine. In the same way, personalized precision medicine, personalized herbal medicines, we are planning for that. So one plant will be suitable for one person, because the human genome project, because from that we are able to identify the genes responsible, what are the plants and what are the enzymes receptors that are supporting you, as well as we are also having the plant database. By integrating both the thing, we can have a personalized herbal medicine that can personally work for you, not for everyone. Now here we need collaborations, huge collaborations. Why? Because in the AI assisted or machine learning, we are not going for a CSC department or a mathematician to make a formula and everything. So as well as we are not here to identify the plants and other things. So the pharmacognostics or a botanist will identify it, and as a pharmacologist, we are now going to work out on the model development. And the CSC department, we are giving our objectives to make it as a model, and all this togetherly come out with a product. Instead of working in an independent, you work with a group so that a modern validation parameter can come onto the market. So now this can create an empirical knowledge and into modern validation. Imagine this is the real world science. Now there is a huge demand for herbal medicine having application of machine learning. And the only thing is that we need to create suitable data. How much data you can collect it, based on that availability on the objectives and points, we can design the drug. Now going for the next slide, drugs. What we expect as a pharmacologist, drugs are going to binding to the target proteins, and we are going to solve or cure the disease. Irrespective of that, sometimes the drug may also bind to the off target or other than the receptor or other sites where it is showing in other side effects or adverse effects. Sometimes it may bind to the some other receptors, or it can also cause a what you call reverse pharmacology, where it is binding to the other areas and it is showing some different kinds of action. So it can also say that drug repurposing, where it can bind to some other area. A very good example is metformin. Normally we use the metformin for diabetes, but the metformin is also very good useful for your cancer. There are a bunch of drugs that are useful for repurposing, the different purpose of the thing. Okay, so now we doesn't know exactly how the drugs are going to the site, how they are functioning out, and how they are working out. So now only thing is that we need to identify there is a multi-component, the pathways are different. We have to identify that pathway. If we can identify, it is easy. Even the allopathic drugs, what we are using till now, we only know the one side that is a curable disease. The other part where it is binding and what could be the adverse side effects, possible side effects, and future side effects, or future what you call chronic pharmacology or chronotoxicology, we doesn't know. So it's a toughest part for us to identify these things. So in the similar way, if you go for the next one, what we expect is that the drugs are directly binding to the receptor site. Next slide. Next, directly binding to the site, and they are showing the actions. The basic drug target regulatory target network will be there. But irrespective of that, what is exactly happening? We expect the things like drug is binding to the receptors, and it is showing the next side effects, and it is showing, and all it is activating the receptors, and your drug disease is treated. Irrespective of that, the steps are not forward further. Why it is not forward? Where it is missing out? So now here comes our network pharmacology. Where we are missing, step by step, each steps are missing. Why can't we validate before it enter into the in vivo studies, so that our load and workload can be decreased there? So here we are clearly able to express how you are going to express. Now you see, now there is a yellow color line. It can clearly show how it is showing the action irrespective of the cross marks. It is blocking the signal and it is showing the negative effect. So all these things can be easily identified, and we can predict the toxicology as well as the pharmacology of any herbal drug, irrespective of the drug. So this is how. So that's the reason we need a network pharmacology for assisting the drug directly to the site, and we can also expect the toxicology also, because as it is binding, we can also identify where it is binding, different locations other than our expected receptors. So you can predict the toxicology and metabolomics also. So that's an important thing. That's why we are supposed to use the network pharmacology before going for the animal studies. Next slide. So whenever you are going for the docking studies, where that's a docking studies is one of the integrated part of the network pharmacology. That's the third step that we'll be using for your thing. So in the docking studies, what you're going, you're preparing the receptor and the binding site detection you will be doing, and preparing the docking inputs. If you want to add hydrogens and other things, you are using your chemistry people for preparation of this docking. And you are going to do the experimental validation via in vivo or in vitro studies. So whatever the receptor is there, you are making the pocket detection tools, conformational change, how the protein should be there, how the proteins can easily enter into the site, and the ligand preparation, and how you're going to hit it, all the things you will be doing. So these ligand based preparation will be 2D molecular based, that the structures are available from the PubMed. You're making a small conformational changes, and the proteins are also there. For the proteins also, you're making the structural changes so that how the drug can easily bind into that in the pocket to have the effective action. So now the docking will be there, and after the docking, the best scoring, the lowest energy binding compound will be suited, and that will be chosen for your drug likeliness. That are the hit compounds that can be screened on your animals, right? Now let us go for a real-time experiment how we performed here. So we know that in the diabetes mellitus, we have the type 1, type 2 as well as the type 3 diabetes. So one of the major cause of insulin resistance is abnormal serine phosphorylation. Next slide. Of the insulin receptors. So in the insulin receptor downstream signaling, you can see here it's a mechanistic pathway here. In normal condition, the insulin is binding to the insulin receptors and it is activating the IRS pathway, that is insulin receptors, and via tyrosine phosphorylation. Remember this name, that is tyrosine phosphorylation. This tyrosine phosphorylation is indirectly activating the P13 pathway as well as AKT pathway. This AKT pathway indirectly is activating your GLUT translocation. What is in the GLUT? So GLUT are the receptors that transport the glucose molecules into the cell. Now that's the thing, if the cell cannot utilize the glucose, it will die. So now the GLUT receptors or the glucose uptake can be activated here. But what's happening in the type 2 diabetes, where insulin resistance or signaling is lost, where the IRS, that is serine phosphorylation, is completely disrupted. Because of the disruption of the IRS pathway, the P13 and AKT pathway, and glucose uptake is reduced. So impaired glucose signaling and finally is causing decreased glucose uptake, and it is causing hyperglycemia, and that's the reason for insulin resistance. So now our target is targeting the proteins that are involved in dysregulation, provides therapeutic strategies for diabetes management. So now in your inflammatory conditions or any other stress conditions, IRS undergoes serine phosphorylation at multiple sites. So this abnormal serine phosphorylation impairs IRNS functions, inhibit P13 AKT pathway, and leads to the insulin resistance. That is going to cause the reduced glucose uptake. Now this is our hypothesis for this thing. So IRS pathway and translocation of the GLUT, so that the insulin resistance can be overcome. So this is a long-term strategy for us. Now we took one of the hypotheses as we are activating this IRS pathway or serine phosphorylation we are going to activate. So now what are the sources of phytoconstituents? In the next slide, if you can see, we have various compounds like flavonoids, steroids, phytochemicals, carotenoids, and terpenoids, and all other compounds. Every other compound works in a different mechanism and different locations for your insulin mechanism. So some of the plant may work in your liver, some of them will help in the DPP4, some of them works in the pancreas, and some of them uptake in the peripheral glucose uptake. So we doesn't know how it's going to work and what plants are going to think. which one is going to work effectively in which area. So it's a multi-complex system. The plants are the multi-complex system and the diseases are the multi-complex system. Now how you are going to integrate, everything is working out. If you see individually, if you see any paper in your articles, all the things are showing that it's showing very good activity. Now the question comes that how does it work? How can we integrate this one so that to show a best mechanism of action? The same plant or the same leaf extract having these multiple components, and for the multi component we are going to isolate these compounds and we are going to show it up how it can be used. So if you go for the next slide, we use the network pharmacology here. Now we have selected one of the plant. In the one of the plant, we have many biomedical products that are available in the plant. So now we have to target the proteins. What are those targeted proteins? We doesn't know. As a normal ethnopharmacology, still now since few years only the network pharmacology has come into the picture, and we have to identify the targets. The targets can be identified by various softwares and other things, and we are going to construct it. Network. Now, if you see the disease is there in the gene, and we have identified the targets and the compounds. Now, the compounds will be activating or directing for the different targets, and finally, we can be cured for the disease. So now, example, you see now this is the key pathways that we expected in our pathway. That is activating the P13 AKT signaling pathway, glucose metabolism, insulin signaling, and inflammatory response. Now, this network pharmacology is a holistic approach. Why? Because it is a multi-component, multiple compounds are present, multiple bioactive compounds are there, and multiple targets are there, multiple pathways are there. So we are not directing into the single pathway, we are targeting on the multiple pathways. Now, from that, we are simplify into the key targets, that is, what are the key pathways that are responsible for this plant. And now we are able to understand how we can establish the mechanism of action of the natural products. Still now, because before the network pharmacology, it is very hard to create in pharmacology what is mechanism of action of the herbal drugs. But by using the network pharmacology, guys, we can identify and create a mechanism of action of the product. And this can be useful for discovery and effectiveness therapeutic thing. So therapeutic effectiveness can be improvised, and by ADMET studies and other pathways, you can also predict the LD50 of the products also. So network pharmacology provides a systems level understanding how natural compounds can modulate into complex diseases like diabetes. So we will be utilizing that. We are having some of the products, we will be using that. Now what does exactly there in the network pharmacology? In the network pharmacology, in the next slide, you will be seeing you have an integration of the genes. Already the human genome project is there. You are in the genes, the human related genes are there, and the gene expression can be studied, as well as the proteomics are also there. Some of the students after the final year will be going for the bioinformatics, where the genomic studies and proteomic studies will be integrated in their studies, in their projects and other things. Proteomics, we are able to identify the protein targets and the protein level of expression that are present inside, or it might be present on the molecular basis. Along with that, we are also having the system biology, where you are going to elucidate the pathways, how the drugs are going to cause the pathway, how my herb, how my drug, plant species are going to show the mechanism, expected mechanism of action, expression of each protein, whether it is having the metabolic or it is having the cellular component or it is expressing the genes, everything can be expressed by our network pharmacology. And because it is a network, it is a multi-component, we need a visualizing techniques. Because by all these things, you see it is a web of things, web of mechanism, web of links for all the proteins, and we can't easily identify which one is responsible and how it is causing. And next one is data science. Data science meaning is how much data you are going to show to give to the computer, the amount of changes that are there, and prediction of the compounds can be very much easy. And visualization can be done, how easily it can identify. And finally, the key targets for treating can be done. So now, as part of the diabetes mellitus and my plant is constrained, we are going to identify the insulin signaling, inflammation, and how it is going to cause angiogenesis, and along with that, how it is going to control the glucose and wound healing. So these are my main objectives, and how I am going to utilize the network pharmacology can be screened by step in the next mechanism. Now, in the next slide, if you see, what I have done is very interesting thing. Uh, we have done soft slate extraction, and after extraction, we have given the sample to the LCMS, to the GITAM School of Pharmacy, Vizag, where our analyst has created GCMS MS samples were done. Among the GCMS samples, the top 10 compounds with highest retention time and high peaks and high intensity compounds were selected. Next slide, sir. So now in that compounds, we have identified many compounds like vitexin, neuroclonogenic, isoquercetin, hyperoside, chlorogenic acid, pinoracil, rosmarinic acid, penodine. These 10 compounds, because these 10 compounds are showing the high peak intensity and high availability. So there are multi compounds. Here we are able to find almost like 1500 compounds were there. In the 1500 compounds, we chosen the highest peak available compounds only, not all the compounds, only highest availability, because each plant will be showing availability as well. We have to choose that plant in that context as well. So now our plant was chosen in this way. So now among these plants, we have identified the structures by the PubMed. Whenever the GCMS data is there, we have based on the PubMed data and other thing, the similarity compounds were identified. And interesting, few of the unknown compounds were also identified with the structures were there, but we could not predict the names and other things where it is not established in the PubMed and other softwares. So from the available data, these are the established products that are there from the PubMed. So after that, what we did it. So now prediction of the compound and the disease target. Now the interesting slide comes in the next one. So now the top 10 compounds that what I have selected, that we have given into the Swiss ADME. Now, so Swiss ADME, just you can give the canonical forms or the structure, that C double bond C, CHH, that is called as the canonical forms. And from that, we have identified the compound targets, because based on the structure where it can bind effectively, that can be visualized from Swiss ADME. And next one is type 2 genes. Now, what are the genes responsible for causing the type 2 diabetes? We can get this data from the gene cards. From the gene cards, based on the availability of the compounds, which compounds can be targeted to these genes can be identified via gene cards. In the gene cards, we have 3 species, human specimens, humans, rats, and rabbits data can be generated. So now, by using the Venn diagram, what are the genes that are common for the plant as well as for the genes we have matched. Now, human genomics projects integrated into our study. So 10 plant products and genes we have integrated. And now what are the genes that can be common for both the plant species as well as genes we have expressed in terms of Venni, that is a software tool. Guys, remember, visualization is also important. How much data you do, how you present the data is important, because as this is the plant extracts as well as germline project, we have a huge data here where we can't easily identify that. So a Venn diagram is useful where you can identify a common diagram. Common targets can be identified. Next. Now we use the string. Now string. So here, whatever the proteins that are there, here in the string, we will be able to identify the protein-protein interaction as well as Kyoto bar charts. How? Because it will be each plant extract on each specimen will be identified, and it can target any kind of receptor it can express. We don't know how we are going to express, as well as gene ontology, whether it is expressing the cellular, metabolomics, and other things. All these things can be seen by a simple software that is called as a string. This string software, if you can apply that, we can have a network of protein. How many proteins are there, how they are interacting, which proteins are targeted, which proteins can be targeted, which are the highest targeted proteins or interrelated with this plant product can be isolated. From the KEGG bar charts, we can identify what kind of molecular mechanism or the biochemistry it is involved in that. In the gene ontology, what are the genes that is this drug can this drug can act on these genes about the diabetes. So this is an interesting software that we can use. It is available online. All these versions are available for free. You can sit in the home or in the college and you can use this softwares for predicting your models. Only thing is that get the LCMS data or GCMS data and you can predict the molecular mechanism of your plant. Your BPharm projects and MPharm pharmacology projects can be done by sitting on your computer. After that, after expressing the molecular mechanism, a simple in vivo studies or in vitro studies will be suitable for submission of the projects. So if you go for the next one, after that we have used the cytoscape. In the cytoscape, we will be using all whatever the compounds are there. Now we have chosen the 10 compounds, and now from the cytoscape we will be able to identify the targeted proteins. In the string, the list of compounds around 140 or 150 compounds can be, all the proteins are interlinked with each other. But the cytoscape help us very easily to what are the target compounds that are come and which is the one which are having the highest binding affinity. So now this is the where we kept into the string all the compounds that we have uploaded into the cytoscape, and from that we can easily identify. Now we have gone for the next thing that is called as cytohubba. Next slide. In the cytohubba, all the proteins are uploaded. Whatever the proteins you are thinking that it is interlinking, all the proteins are uploaded, and from the uploaded we have the red color one are the one which are having the highest interaction. And in the interaction also the top 2 priorities will be given. We can't work it out for the whole complete thing of proteins. So here the STAT3 and CASP3, now we got these are the 2 responsible for causing this thing. Now as we got the receptors, now in the previous days we directly used to target any kind of enzyme or the receptor and we are trying to do the docking. Now no need to do that directly. Go for network pharmacology, find out the receptors are the targets, then you dock it. Irrespective of blindly docking any kind of receptors and other things, enzymes and other things, first find out which one is, this will be a highly relevant and it will be a highly precision for you. So now whatever the 2 proteins are there, we are going to dock it by Autodock software or by MolSoft software. We could dock it with these 2 proteins, and based on the docking score, the lowest score, we will be choosing and filtering that compound. Now here we got STAT3 and CASP3. Now STAT3 and CASP3 are the enzymes that will be responsible for autophosphorylation and tyrosine phosphorylation compounds of our insulin resistance. Next slide. Now, if someone asks what are the canonical forms, as a pharmacology people, we doesn't know the canonical forms. Write down, this is the canonical forms of Vitexin and the top 10 compounds. So these canonical forms can be available from the PubMed. When you easily identify and know the names, the canonical forms and structures can be easily downloaded from the PubMed. So these are the 10 compounds that we should selected for our study. Next. Now we have applied the ADMET. In the ADMET studies, we have able to identify the phyto targets. Now 1000 compounds are there, and we removed it, and finally we got the targets of 475 by ADMET. So this is the query molecules and how they are finding. Based on all these things, we have got unique compounds, and we finally got the 475. This is our results that we have showing from this ADMET, Swiss ADME analysis. Right. Next one, we applied the gene targets. Now what is this gene target or gene cards? So in the gene targets, we got 21,351. Total gene targets we got is 21,000. Now if I apply by removing the duplicates and the relevant score, hello? We have identified. Yeah.
Hello.
Yeah. Hello.
Admit करा म्हणलेत का?
Okay, we got the gene targets of 21,359. From that, we got the relevance score, and we got the unique targets of 1262. Sir, go for the next one. That is the gene gene cards. So in the human genome database, so now this is the gene cards that is integrated database, user friendly, and we can predict the human genes. So now it is automatically integrate your genes, and almost like 200 websites uses genomics, transcriptomes, proteomics, genetics, clinicals, and functional information. All the things it will integrate. And finally, we got the unique targets of 1262 targets we have identified. Next one. So now this is the Venn diagram for you. In the Venn diagram, so now we got 1000 genes, and from our plant, that is Laxiflora, 326 genes that are responsible for diabetes, and these are the diabetes genes, and we interrelated with these things, and we thought 149 are the common genes that are responsible. Now these 149 genes or these things, how plants and other things, how they are correlating, we have established in the presence of protein interaction via string. So we established all these things in the string, and you see it's a mesh of things, all are interrelated. We can't easily separate them, which one is interacting more and other things. Now by using the other softwares, by using the KEGG pathway, next one, by using the KEGG pathway, you can see here these plants are activating the different mechanisms like EGFR tyrosine kinase, hepatitis, proteoglycans, cancer, prostate cancer, endocrine resistance, small cell lung cancer, human cytomegalovirus, and HF signaling pathway. So my plant is able to inhibit these things. Now, if you see the green color with highest thing, these are the genes that are dominant, and my plant is able to inhibit these pathways in the cancer. One of the mechanism was established how it is able to cause the pathways. Now, if you go for the gene ontology, here you can see various pathways also. MAPK pathway, cellular response, autophosphorylation, and orgonitrogen compound, as well as cellular response to oxygen containing compound. Some kind of antioxidant activity can also be expressed by this. We can establish one of the mechanism from this one. So the circle, the bigger one, the action, or it is a more relevant one, that is a gene count. Next one. Now, molecular function and gene ontology. Here you can see directly into the biochemical pathways. Now, if you see the protein kinases having a bigger circle with the green color one, as well as alcohol group acceptor, phosphotransperases, as well as protein kinases, as well as tyrokinase activities. So now these are the things that are related. Now we also found out the mechanisms as well. Molecular mechanisms is also established. That's an interesting thing. Next one. Now, by using the cytohubba, as I said, so these are the various proteins, and from the various proteins, we have identified only top two compounds that are interacting high, that is the STAT3 and CASP3. Okay, hope it is clear till now. From the top two compounds, we have identified two. Now, the next one comes is the docking score. So now, by these two proteins, we have docked all the 10 compounds. And from the 10 compounds, we have identified Vetexin, as well as SD36 stat and dicafeoquinic acid. These are STAT3 proteins that are binding with the STAT3. So now we are also done with the Autodoc software, and we established Vitaxin and SD36 and dicafeoquinic acid is having this total energy of binding capacity that we are able to establish. Along with that, we also done with the caspase-3. So in the caspase-3, we could identify isoquietin, rosmarinic acid. Next slide. Rosmarinic acid and hyperoxide. So here also we did the same thing, and we could able to identify the pockets where our drug can bind into that. So now this is till now, I think it is established till here. Now the next question comes is how we are going to establish the mechanism. Next slide. Yeah, this is the signaling pathway where there is, if there is a more amount of inflammatory cytokine response, where in the JAK pathway, the STAT3 expression will be there. Because of this, Vitex band to the STAT3 and modulate the activation and downstream activation of SOC3 upregulation. This SOC3 upregulation is going to inhibit ironase pathway and APT signaling, and it can also causes the glucose uptake. So now by binding our Vitaxin, it is normalizing the SOC3 as well as restoring your IRS and enhancing the GLUT4 and improvising the glucose uptake and insulin sensitivity. So now this modulation of the STAT3 by Vitexin helps in insulin resistance, enhancing the glucose uptake, and may elevate insulin resistance in type 2 diabetes. This is one of the answer for what you call STAT3, how it is working on my Vitexin. So, we got a Vitexin as well as the STAT3. Next one is if you are going for the Cas3. So, whenever the glucose uptake is there, mitochondrial dysfunction, and it is causing the release of cytochrome C from your endoplasmic reticulum. This endoplasmic reticulum, the cytoplasm, it is activating the FAP and caspase 9, and it is causing the beta cell apoptosis. This is the mechanism. Now, what is this isocoatin is causing? Isocoatin is modulating the Cas3 and reducing the Cas3 activating and reducing the beta cell apoptosis, and helps to preservation of beta cell mass function, and it is causing improvised insulin secretion and glucose homeostasis. So, now we have established the mechanism of the model. We did not use any laboratory till now. We did not use any animals till now. We just identified the 2 compounds and 2 target, 10 compounds and 2 targets, and we established the mechanism. So, now after that we are supposed to do with the in vitro studies of the respective compounds. We are going to plan for SDS phase and gene expression studies also for both the compounds. Next slide. So, now in the drug discovery phase, everybody know there is an early drug development, and from that we are going to identify the lead compounds. 20,000 compounds will be there, from that 1 billion, and next we are going to do the in vitro, in vivo, and 250 compounds from that 0.5 billion, and we are going to study the safety studies, the clinical trials, safety and efficacy from that 5 compounds approximately, and finally 1 compound will go for the phase 4, that is approval and manufacturing phase. So, we are taking almost like 10 to 12 years of drug approval phases. Now, by using the network pharmacology and other things, now this term can be minimized from 3 to 6 years, can be minimized here by using the network pharmacological studies. Most of the companies are approaching the using of network pharmacological studies. Instead of having the blind high throughput screening and other techniques, we are using these things. Next slide. Now, how you are going to use these machine learnings? So, these machine learnings can be used for disease diagnosis, predictive analysis, drug discovery, disease surveillance, and medical device development. So, machine learning, now the integration of the pharma people with the CSC people is very much important. For the next generation, if you want to survive into the science and research, integrating the pharma people with the machine learning or the CSC people. CSC people have the knowledge of how to code and write the algorithm for a disease. If you can collect the data and give the prior objectives of your data, we can easily predict a model and create a model, create an algorithm with the help of the CSC faculty or CSC scientists, and we can predict any kind of disease that can be very much useful to our predicting. Example, one of our students, they have developed one model for the skin cancer. By directly taking the photograph of a sore on the skin, they can easily identify whether it's a cancer or not, and what stage of cancer is there. So, we are collaborating with the CSC department. So, now here the question comes, the pool of data should be generated. They are supposed to collect a large pool of data, and based on the color complexity and other things, we are going to make it a language for the machine to learn it, and finally we are integrating and making as an app. That app, by directly selecting on that, they can easily identify the disease. And we have also working on non-alcoholic fatty liver diseases also, how it is working up by the help of CSE. Now, the question comes with the PharmD people is how well you are able to identify and collect the data and make a key objectives and what you call diagnostic criteria, how you are diagnosing the disease. Here we need a skilled people where we can identify it. Next one, deep learning. So, now whenever you are doing into the deep, any kind of learn any one single technique into the deep knowledge, and you can go with the diagnostic, that is a medical imaging. You can identify the disease and classify it very easily, and the treatment can be made personalized, drug discovery, segmentation, and we are going to get healthcare operations and management will be very easy for us. In the irrespective of spending crores of money on healthcare, small technique that can be easily be done. Yeah, next one. So in the machine learning techniques, there are various softwares and that we will be using. As a biology people, we doesn't know about the softwares and other central neural networking techniques, JVM, forest, random forest, and other kind of things are there. So now those people are replacing our studies for generating the models. Now the question comes is that how, when we are going to integrate with them or when they are collaborate with them by using these new tools, they can easily identify it. They are ready to generate the algorithm for us. The question is that when you are going to bring the data with them, they are ready to generate it. Now the question, we have to go with them, come and collect the data, give the data, help them in diagnosing and identifying, explain the pathophysiology, identify the crucial points, and they will generate your algorithm. So please make free to collaborate with other CSC and other departments, machine learning or artificial intelligence people. They will be really helping you to generate a new model or algorithm for you. Next slide. So irrespective of saying all these things, let me show a small example where it is published, the data is published. Here you see the scientists or PharmD people collected a normal Alzheimer's disease MRI scans. So in the MRI images, they have collected the brain images, brain on the nodes and the structure and architecture, they have loaded into your machine learning things. And based on the symptoms, mild dementia, moderate dementia, and severe dementia, so they have identified the patients and made the machine to learn more, deep learning, more and more. And finally, they have come out with the software. Now, by directly without touching the radiologist, by seeing the things and features, the model will say what stage of Alzheimer's disease the patient is suffering with by seeing the MRI image. So now the success rate for this is 92% of the success rate can be seen with this imaging. So those this is the published data elsewhere. So they are using this kind of machine learning tools for identifying and diagnosing the disease. Next slide. Along with that, they have also done one of the studies that is Indian Institute of Science and in collaboration with AIMS Rishikesh. So now what they have done, they have identified the seizure patterns. So now how the normal seizure patterns will be there based on your ECG and encephalograms, and what are the partial seizures, what are the generalized seizures, atopic seizures, absent seizures. So now based on the ictic phase, they will able to identify what are the peaks that are coming out and how the seizure prediction can be done. So this is an interesting thing that is done and it is yet to be published. That is Indian Institute of Science and Rishikesh has worked on this concept. Now imagine how they are collaborating, Indian Institute of Science and AIMS Rishikesh. So now where we are in between Hyderabad. Okay, we can collaborate with other sciences to do this kind of work out. Next slide. Now, the only thing is that we have the large sample data collection of medical history imaging. You as a PharmD students, we are able to collect the large amount of data and genetic information, ensuring the data is stored, structured and formatted. You have to store exactly in that and make it as a spreadsheet. And irrelevant data can be removed, and you can able to missing values and other things, you can add it up for the spreadsheet, and any outliers or anomalies are there that is affecting the data analysis, removing, that is called as a data cleaning. And now it should be normalized. So it should be normalized, choose the appropriate normalizing technique that is called Z-score. That can be done by the CSC people, no need to worry. So apply the normalized technique and numerical values, and from that extract the features that are unique, gather, and utilize the domain and knowledge data, apply these techniques and to reduce the dimension of the study. So now by all these things, you can create one new model. Right, as a next generation students are there and faculty are there. Now the DNA sequence is available. Next slide, sir. DNA sequence is available, lifestyle thing is available, gene expression data is available, imaging is imaging diagnostic tools are available, and patient phenotype or patient history is available. Now all these multi-dimensional studies can be integrated into your data sets. If you can integrate, then make the system or the machine to learn it. Machine learning, make it to learn, create an algorithm. How much data you give, the machine will be able to learn that. After learning that, it will able to show visualization, and you can easily analyze it. The system will able to analyze, the AI will able to analyze it. So the success rate till now is around 70 to 80% of the success rate can be seen if you have a sufficient amount of data. And from that, till now, the personalized medicine can also be done by integrating your drugs with this thing. Yes, now how you are going to integrate your personalized medicine here? Okay, next one. So in the patient, we have the whole genome and select the algorithms, gene functionalities, and whatever the genes integrated, what are the drugs that are there, integrate with the both, and you'll have a drug dose and improved therapeutic effect can be done. The first choice of drug for a personal drug can be done by the personalized drugs. So now by the human genomic project are approaching, you can take the human genome and the drugs available, match with that, the best match can be given to the patient. So that is called personalized medicine based on your genes. So now what we thought is that the patient, a problem often set those one size small fits to all approach. We say that one drug is working for all the patients. No, it's very tough. So we have to go with individually. In our regular cases, the medication may stop or dose can be lower, dose can be increased. We doesn't know what exactly. So based on the gene expression and genomic studies, we can easily identify it. So next slide. So if any doubts, please let me know. I'm happy to help you. Any doubts?
Participants, please, if you have any queries, please post that in the chat box. Or you can unmute yourself and you can ask directly.
Yeah, I think. Yeah, Pradipti, with convergence of generative AI, multiomics, and ethnopharmacology, are you approaching a future where AI can, design new traditional drug molecules, synthetic analogs inspired by traditional wisdom, but optimized for a
Okay.
specific genome project, closing the loop between ancient
Okay.
knowledge and futuristic precision?
No
Yeah, it's a good,
program.
very good. Yeah, it's a. I really like this, Pradipti, having
Program is
a.
over here.
Yeah, it's a very good thing. Yeah,
Yeah.
AI can predict new molecules as well, because we have a
Okay.
lot of
Okay.
data now. Whatever the machine is able to give, based on that, it will be showing it. How give small projects to B Farm students? It's a small project because it doesn't take much time. Hardly you can take 1 to 2 months.
Sir, we
If
have
you,
a question in the chat box.
yes, sir, I'm answering to that. Which databases are available? Madam, databases are available for all the diseases. Human Genome Project is done. All the databases is available for breast cancer as well. We can work it out on that. Now a small project we have done for the last 2 months for the diabetes. We have worked on this one. We can work. Yeah. How can we start and initiate with our bachelor students? Yes, bachelor students, they can extract the plant sample. They can go for the GCMS or LCMS, get the plant extract, go for network pharmacology, integrate. You will have a preliminary data, how the drugs are going to work in a molecular mechanism. Yeah, we can go for the B-Farm project also. Our B-Farm students are doing the same projects in our college as well. So they are able to understand it. It's a multi-complex, but how well you are going to insist or give the knowledge to the students, make it a different. We have large pool of softwares that are available online. You can utilize that and you can explore your plants. Number of plants you can use, the data, and you can get number of papers from that. Large pharma historically exploit traditional knowledge without benefiting share. Yes. Can blockchain integrated AI systems can be designed to create immutable provenance trials that automatically trigger benefit sharing, smart contracts when a traditional remedy becomes a commercial drug? Yes, these pharma companies, I know, we know, they will not suggest us to go for these herbal drugs, but to overcome the side effects and adverse effects and everything we are going to create. We should be coming that for the patient safety is the first priority rather than of economic things. How can we learn such types of tools and keep updated? Sir, even I too don't know. When we attended one of the FDP, I came across this network pharmacology. I immediately started learning this network pharmacology concepts and saw a few papers, how they are doing. Starting this, we were worried how to intercut, but connecting the people, so you can connect me back for any kind of queries and other things. We can work together, and we will be assisting the students also. We are planning to have a workshop on network pharmacology in the coming days. So you can welcome there, and we will be having a hands-on training for you. But only thing is that you should get your LCMS data prior. If you have the LCMS data of your plant, so we can come out here where we will have a one-day workshop here. We will plan a hands-on training on your respective plants. By attending the FDPs, you have to gain the knowledge every time. Kushi, thank you. How can we determine which constants are effective for breast cancer? Now that's the thing. Many components are available. Now, what we know as a pharmacognosy and ethnopharmacology, we think the flavonoids or phenols based on the structure, these are best suitable. So, irrespective of that, let us go or deep into that. We can't directly say that flavonoids or other phytoconstants are responsible. Dig into that, the genes that are responsible, we can establish a mechanism, and we can easily identify which one is best suiting for your cancer. So because cancer is a well explored, genetically, proteomics, metabolomics, everything is well explored in the cancer. Now, only thing is that you have this 10 phytoconstants of your plant extract. You can mix up with all the softwares, you can do it. Yeah, Dr. Kushi, thank you. Praditi, a remedy was used in Ayurvedic practitioners in Telangana may have phenotypically similar used in Amazonia and Africa traditionally. Are cross-cultural pharmacological convergence algorithm being deployed to flag such parallel potential revealing universal mechanism of action? Yeah, it's a very good, interesting question, Deepthi. Yeah, we can establish a universal mechanism for our plants. Universal mechanism of action of the plants. The same plant can be used in China, traditional Chinese medicine, or allopathy, Ayurveda, Unani, Siddha. Now, if you can mix it up, we can make it up. Or else herbal or ethnopharmacology, it's very tough for us for the survey because we have a problem with establishing the mechanism of action. We can't extract the drug because when you extract the purified compound, the action of the compound may be lost. When you take the whole plant, the drug will be there. How it was suggested in the monographs and other things, you have to take in that. Instead of that, if you can take it in the identify in the simplest form like allopathy, like paracetamol or ibuprofen, and giving the patient, sorry, it should not be there. It should be given wholly, completely. Don't go for isolation of the individual plant. For establishing the mechanism of action, you can do, but taking the complete plan, you have to take a complete plan for the treatment. We don't know because one of the drug may acts, different components will act as a different. Some of them will be acting as excipients or the what do you call other kind of thing. Everything will be working together if a plant is taken wholly. Don't take it as an individual. From my suggestion, if you take individually, the drug doesn't work. If you want to separate a curcuminides from curcuma, it doesn't work. The curcuminides are 3 compounds. If you want to separate it, no, it won't work. You have to take it as a wholly. Then its functioning is good. It is already established model. Why you want to separate it separately? Yeah, Deepika Prajapati, thanks. And sir, next slide. Hope there is no questions. Yeah, these are my references that we used. And thank you MLR College of Pharmacy for providing this opportunity, as well as thank you GITAM School of Pharmacy. And hope we will work together. You can collect my mail ID from the GITAM website. You can contact me for future sessions as well. I'll be ready to assist you. And in future, we are planning for an FDP on hand of planning these things of network pharmacology. We are exploring the things, and we'll plan a FDP where you can come and utilize these sources. You can do your own work here. You don't need of any softwares. All the softwares are available. Only thing is that how you are going to integrate all the softwares together to work out. Learn these things. B Farm projects can be done very easily, and you can say that how my plants are working out very easily instead of going for all these things, all allopathic system of medicine and other things. You can also do the clinical trials. If the mechanism is established, herbal drugs are the super drugs that can be used. Yeah, thank you, thank you, sir, and thank you, Saujanya, and thank you, Sultana, madam, for providing this opportunity, and thanks a lot. Thank you.
Yes, sir. Thank you, Vinyas, sir. You just please be online for a few more minutes.
Thank you, Asra. Yeah, we will be working out. You can just reach me out from my website or by directly from the GITAM website, GITAM School of Pharmacy. We can work it out. We can collaborate in that. Yeah, we will be sharing the PPT as well, Pooja Yadav. We will be sharing the PGT. Thank you, Pradipti. And that's a very interesting question from Pradipti. Yeah, Saujani madam, you're back.
Thank you, sir, for your wonderful session.
Yeah. Thank you, madam. Thank you for
So now I
providing
would
this
like to
opportunity.
invite Dr. Suvendra Saha, introduce a guest of honor for valedictory program and vote of thanks for FDP.
Thank you, Dakshina. Thank you, Anamika. Rajeshwari and Pooja, thank you. Yeah, Pradipti, yeah, I will share that. You can collect the details from Dr. Saujanya. Dr. Saujanya is available from MLR College. She will be sharing my details and contact details. Thank you, Pradipti. Thank you, Rajesh and Mustaq, for listening my session. Thank you, Srimukhi. Thank you, Dr. Jain Mukherjee. Thank you, Ragini and Sandeep Patil. Sorry, Ragineni and Sandeep Patil. Thank you, Justin and Sangeeta Madam. Dr. Kushi, thank you. Augustine, thank you.
Good afternoon, ma'am. Good morning, Dr. Shahida, ma'am. Hello, ma'am.
Hello, good morning. How are you?
Good morning, Dr. Shaila ma'am.
Yes, very good morning. Are you able to hear me?
I am Dr. Subhendu Sahu, Professor, Department of Pharmacology, and Academic Coordinator of Mari Lakshman Reddy Institute of Pharmacy. I take great privilege in introducing our esteemed guest of honor for valedictory programs, Dr. S.M. Sahida ma'am. Dr. S.M. Sahida ma'am, M.Pharm PhD, MBA, is an accomplished academician and researcher in the field of pharmaceutical sciences. She is currently serving as an associate professor at Crescent School of Pharmacy, B.S. Abdur Rahman Crescent Institute of Science and Technology, Tamil Nadu, and has over 2 decades of reach, teaching, research, and administrative experience. She has previously held key academic positions, including professor and deputy dean, student affairs, contributing significantly to academic planning, accreditations process such as NAC and NBA, and student development initiatives. Dr. Saida has successfully completed several funded research projects supported by DST, UGC, SERB, and ICMR. She holds multiple national and international patents, including innovations in drug delivery systems, artificial intelligence in healthcare, and herbal formulations. With an impressive publications record of over 40 research papers in reputed journals, along with book chapters and conference proceedings, her research interests include pharmacognosy, phytopharmaceuticals, nanotechnology-based drug delivery, and pharmacological screening. She is a recipient of several prestigious awards, including the National Best Domain Scientist Award and Best Technical Teacher Award. She is also actively involved in editorial roles and professional bodies such as APTI, IPGA, and IACP. Dr. Saida has guided numerous undergraduate and postgraduate students and is currently supervising PhD scholars, reflecting her strong commitment to academic excellence and research mentorship. We are honored to have such a distinguished personality as the guest of honor for this EFDP program. Now, I humbly invite Dr. S.M. Sahida to address the gathering and share few words about the program. Ma'am, we would be grateful to hear your valuable insights. Thank you,
Thank you
ma'am.
very
Over
much,
to you, ma'am.
Dr. Sahu. Yeah, thank you very much, Dr. Sahu, for your, very detailed introduction about me. It was a privilege for me. Thank you so much. And as I start to the guest of honor, good morning to one and all present here. Dear faculty members and dear participants of this 5-day faculty development program sponsored by APTI Telangana State Branch on the title Integration of AI and Network Pharmacy in Modern Pharmacological Screening. It is my honor to address the valedictory session of this remarkable EFDP graced by 315 brilliant participants from Mari Lakshmi Reddy Institute of Pharmacy. Over the past 5 days, we have been witnessing inspired sessions on integrating traditional wisdom and modern science, healthcare 5.0, network pharmacology, AI in genomic analysis, and translational ethnopharmacology. These topics are shaping the future of pharmacy and healthcare. To proceed, we should be very thankful to Dr. M. Sunita Reddy, Madam, Dr. J. Shrikant, D. Ashok G. Matani, Dr. Sarvanan, Dr. Krishnamohan Chinala, and Dr. Vinyas Mayasa, whose talk has been very spectacular in giving the knowledge to the participants. I commend the organizing team for the curative and enriching program that fosters innovation, research, and professional growth among the faculty. My appreciation goes to all the speakers for their invaluable sites and to each participant for their enthusiastic engagement and commitment to the excellence in education. This particular FDP will undoubtedly empower educators to inspire future pharmacists and researchers, contributing to advancements in patient care and pharmaceutical sciences. I wholeheartedly wish all the 315 participants their success in their endeavors and hope the learnings from these programs translate into impactful contributions to healthcare. Thank you very much for inviting me to be a part of this memorable event. I extend my wholehearted congratulations to the organizing team, the HOD Department of Pharmacology, Dr. Sandeep Reddy, and the HOD Pharmacy Practice, Dr. Balamurli, Principal Dr. Arunbha, Arunabha Malik, and the whole APTI team and everyone for their successful completion of their EFDP. Thank you so much.
Thank you, madam, for your insightful and inspiring words. Now I'd like to request Sandeep sir to speak few words about ma'am.
Okay. Okay, is it audible?
Yes, Dr. Sandeep.
Good, good afternoon, madam. Thank you very much
Good
for your
afternoon.
message, madam. As a guest of honor, you have given very good message for all of us. And successfully, we have completed our 5-day FDP. On behalf of Mari Lakshmi Reddy Institute of Pharmacy, I take privilege to speak few words. Myself, Dr. Sandeep Reddy, HOD Department of Pharmacology. I thank all the participants for participating effectively for this 5-day FDP. Uh, Integrating of AI and network pharmacology in modern pharmaceutical sciences, pharmacological sciences. And I am very much happy that all the participants have gained some knowledge in relation to AI as per new syllabus which we are going to adopt in the upcoming year as per NEP policy 2020. And I thank uh, to all of, I want to say one more thing, that is, Shaida madam is not a guest of honor. She is my guide, and I completed my PhD from the same university. Thank you very much, madam, for coming as a guest of honor and accepting me.
My
And,
privilege.
and on behalf of Mari Lakshmi Reddy Institute of Pharmacy, I thank management, principal, and my dear staff members and all the organizing committee members for making this event grand successful and completed this 5 days FDP. And thank you very much, all the participants. Thank you very much, Dr. Saha sir.
Thank you, Dr. Sandeep sir, for your valuable message regarding today's valedictory program. Okay, now I am Dr. Sugita, Professor, Department of Pharmacology and Academic Coordinator of Mari Lakshmi Reddy Institute of Pharmacy. Take privilege and honor to propose the vote of thanks on behalf of Mari Lakshmi Reddy Institute of Pharmacy. First and foremost, I extend our heartfelt gratitude to our guest of honor, Dr. S.M. Sahida, Associate Professor, Crescent School of Pharmacy, for gracing these valedictory functions with her presence and for sharing such inspiring closing remarks. Your insights and encouragements will truly motivate us to translate knowledge into impactful educational and research practices. A special note of appreciations to our inaugural chief guest, Dr. M. Sunita Reddy, Principal and Professor JNU University College of Pharmaceutical Sciences, for setting the tone of the FDP with her inspiring and inaugural address that highlighted the significance of bridging traditional wisdom and modern science. We express our sincere thanks to our distinguished research persons for their valuable sessions. Dr. J. Srikant, Assistant Professor, Assistant Dean, Srihar DU, Chennai, Tamil Nadu, for his thought-provoking discussions on the harmony between modern science and traditional wisdom. Dr. Ashok J. Matani, Professor retired, Department of Mechanical Engineering, Government College of Engineering, Jalgaon, for elaborating on Healthcare 5.0 and futuristic research avenues. Dr. Shravanan J., Assistant Professor, Department of Pharmacology, JSS College, OT, for his deep dive into network pharmacology and drug discovery. Dr. Krishna Mohan Chinnala, Professor and Dean, School of Pharmacy, NNRGI, Hyderabad, Telangana, for unfolding the promise of AI-powered genomic analysis, and Dr. Vinyas Mayasa, Assistant Professor, Department of Pharmacology, GITAM School of Pharmacy, Hyderabad, Telangana, for his engaging sessions on AI-enabled translations ethnopharmacology for precision medicines. Each of you has broadened our perspective on how artificial intelligence and network pharmacology can revolutionize drug discovery, patient care, and academic research. Our sincere thanks also go to our principal and convener, Dr. Arunabha Malik, for his constant guidance, leadership, and motivations in organizing this program. We extend warm appreciations to Dr. C.H. Sandeep Reddy, co-convener, and Dr. S. Balamurali Mohan, coordinator, for their dedicating planning, seamless organizations, and tireless efforts behind the scenes. We are also grateful to our collaborators, Associations of Pharmaceutical Teachers of India, APTI, for their support in making this FDP a success. We are delighted to share that e-FDP program received an overwhelming response with more than 300 registrations. A big thank you to all our faculty participants, research scholars, and industry professionals from across the country who joined us on the Microsoft Teams platform. Your enthusiastic participations, thoughtful questions, and active engagement made every session meaningful and interactive. Last but not least, appreciations is due to all the organizing committee members, technical team, and volunteers whose commitment and coordinations ensured the smooth conduct of the event. I would also like to express our sincere gratitude to the management of Mari Lakshman Reddy Institute of Pharmacy for their unwavering support and encouragement in organizing this EFDP. Their visions, commitment to academic excellence, and constant motivations have provided us with the platform to conduct such meaningful and enriching programs. Without their support, this initiative would not have been possible. To conclude, this FDP as a journey of learning, collaborations, and inspirations. Let us carry forward the spirit of innovations and integrations of AI and pharmacology into our teaching, research, and professional practice for a better and smarter future in healthcare. The enthusiastic participations, insightful interactions, and commitment shown by all our participants have truly encouraged us. It motivates us to organize more such programs in the future for the continuous development and empowerment of the teaching fraternity. Thank you all once again, wishing everyone continued success in professional pursuits. Thank you, madam, for gracing this programs, and your valuable thoughts have truly motivated us and added a great value to this EFDP programs. Thank you, madam. We are very grateful for your presence and guidance. And participants, you will all get overall feedback in your registered mail IDs, what you have mentioned while